Thank you, Peter. This is going to be an experimental study, so I don't think if it's going to raise so much participation as the previous clinical studies. Well, during fetal to neonatal transitions, preterm babies have an immature antioxidant defense system and have a structural and functional immaturity of the lung, including surfactant deficiency. Frequently, preterm infants need oxygen supplementation to overcome respiratory insufficiency in the first minutes after birth and even thereafter. And as we have heard before, oxygen supplementation causes oxidative stress, lung damage, and structural and functional alterations of lung surfactant. We hypothesized that if we would precondition mice offsprings with 14% oxygen, which it's similar, gives them a similar PaO2 as they have in utero, and we keep this PaO2 after birth for a while, then we would reduce oxidative stress and surfactant alterations caused by a hyperoxic insult. So we thought that preconditioning these mice to a hypoxic environment for a while, it could also improve their lung adaptation afterwards by keeping their antioxidant potential in a better shape. We designed an ad hoc oxycage, as you can see on the left side. It had two independent chambers, so we could keep a different oxygen concentration in each chamber. We had infrared detectors, so we could know exactly, precisely the moment when the offsprings were born. And we had a special system that allowed us to make changes in the concentration of oxygen in the cage much more rapidly than in the commercial systems. We also had a video recording, because you know that pregnant rats start doing special movements before they start having the babies. And we also put some gloves that allowed the manipulation of the offsprings without changing the FiO2, which is something that we also don't have in the commercial oxy cages. We had three groups. We had a control group up here, which was all the time in room air. Here you have P1, first day, and P7, so we did 2 stages of controls. The experimental group 1 was the normoxic group. Babies were, offsprings were born in room air, and after 8 hours they were injured with 100% oxygen for 1 hour, and they went back to room air. And the experimental group 2, the hypoxic group, was born in 14%. They got also injured with 100% oxygen, and they went back to 21% to room air. We made determinations at the end of day 1 and at the end of day 7. We analyzed the transsulfuration pathway, and we saw that there was reduced glutathione was saved or was spared in this in the group of 14% in the hypoxic group, so it was significantly higher than in the normoxic group or in the controls. When we added some of the transsulfuration pathway metabolites like cysteine, which leads also to the synthesis of glutathione, it was also kept. And we also analyzed the enzymes related with the antioxidant defense system, the catalase, and we also analyzed peroxidases. And they were also, the catalase the activity was enhanced in the 1400 group, in the hypoxic group, and the generation of peroxidases was lower in the 100 to in the hypoxic group. Moreover, when we analyzed in tissue, these determinations were done in lung tissue, we analyzed the markers of protein damage and lipid damage, as you see here, the tyrosines, the oxidized tyrosines, and the nitrotyrosines. They were significantly decreased in the hypoxic group as compared to the normoxic group, both for protein damage and for lipid damage. What was striking is the finding in electron microscopy that, as you see here, the lamellar bodies were highly preserved in pneumocytes type 2 in the hypoxic group, when compared to the normoxic group that received afterwards 100% oxygen. If you see here, the lamellar bodies are these structures, and the amount of surfactant is significantly lower. Here you have higher images of a higher potential, and you see here how the lamellar bodies are perfectly preserved here, or as compared to the normoxic group. We analyzed the synthesis pathway and the transcription factors of surfactant proteins, and if you see here in P1 and P7, the Western blotting showed us an increased amount of surfactant protein B in the group of hypoxic preconditioning, and this was also, we had B-tubulin as the keeping gene, housekeeping gene, and we also saw that STAT3, which is the transcription factor that leads to the synthesis of, among other genes, of surfactant protein B, was also highly expressed in the hypoxic group. When we did the same studies at P7, we found similar results for SPB, but we didn't find that those differences when we were studying the whole transcription factor. So we concluded that perinatal hypoxic preconditioning favors the acquisition of a reducing environment in the lung and a reduction of biomarkers of oxidative stress. Perinatal hypoxic preconditioning increases lung resistance to prolonged hyperoxia, as evidenced by the preservation of the ability to store and synthesize surfactant at P1 and P7. We underscored the relevance of a strict control of oxygen supplementation in premature newborn infants during postnatal stabilization. Thank you very much.